Hello everyone, a very warm welcome to the official page of Read My Course. I am Dr. Shatabdi and together today we will continue with Cell, a Unit of Life. So let's start with our class. So what are the topics we will be covering today? Yes, I know today we are going to start a very important part of cell, that is nucleus. It's a very interesting topic and a bit difficult. So I'll try to explain the basics first in a very pictorial representation, diagrammatic fashion. And so let's start. First is, what are the different parts of a nucleus? We will study about the structures which are found within a nucleus. Next is a little bit of the history of the discovery of nucleus. We had already studied a bit of it. We will study a little bit more of it. Next, we will study an important part, that is what is chromatin and what is chromosomes. Often students get very much confused between these. Even when I was a student, I used to get confused between what is a chromatin, what is a chromosome and all these things. So I'll try to explain it. Then we will come to the different types of chromatin. Then the function of nucleus and the function of its individual parts, we will go in details. And last, we will study a bit about how many nucleus is found per cell, how many nucleolus is found per cell. So let's get into it. See, the diagram at the top is showing a full cell. Okay. Now, when we look at the center of the cell, we are noticing a small blue structure, right? This is the nucleus of the cell. So, when we are looking at the whole nucleus of the cell, it looks something like this, right? Now, let's have a look at what is found inside. Inside we have nucleoplasm. Just like in the cell, inside the cell membrane, we had what? We had cytoplasm. So here, inside the nuclear envelope, we have the nucleoplasm. At the center, we have something which is the nucleolus. Okay, this ball-shaped structure at the center is the nucleolus. Then we have some X-like structures. These are nothing but the chromosomes. They are scattered within the nucleoplasm. They remain freely scattered within the nucleoplasm of a cell. Okay. So with that, we move on to the next slide. So coming directly to the different parts of a cell. Usually, parts of a nucleus. Usually, a nucleus has 4 components. What are they? First is nuclear membrane. That is the outer covering of the nucleus. Next, we have the nucleoplasm, just like the cytoplasm. Then we have the chromatin fibers at the chromosome. And then we have the central part, that is the nucleolus. Okay, so what are the 4 different parts of a nucleus? The 4 parts of a nucleus are, first is the envelope, then is the nucleoplasm, that is the matrix, then we have the nucleolus, and we have the chromatin fibers or the chromosomes. So these are the 4 basic structures which we will notice inside a nucleus, right? Moving on to the next slide. See, when we are trying to magnify a nucleus, or when we are trying to magnify the structures within a nucleus, what do we get? A most important structure is the chromosome. This one. These X-shaped structures are chromosomes. Now, when we magnify the contents in a chromosome, we are seeing these coiled structures, right? These coiled structures are what? They are chromatin threads or fibers. So, when they are in a coiled form, that is the chromosome. When they are uncoiled, that is the chromatin. Coiled is chromosome, uncoiled is chromatin, right? Then when we are magnifying these chromatin threads, what we are getting is DNA. What is it? It is the genetic material which is found inside our nucleus. This DNA, that is deoxyribonucleic acid, forms the basis of all the genetic functions that are occurring within a human body. So the three-dimensional structure of our DNA looks like this. With that, we move to the next slide. So if we are breaking down the DNA structure, we are noticing what? First of all, let me tell you that the DNA is a double helical structure. What do I mean by that? Helix is the shape that it is taking, okay, and double helical because of the presence of 4 kinds of nucleotides. We have 4 nucleotides. I'll tell you the names: A, T, G and C. A for adenine, T for thymine, G for guanine, C for cytosine. A, T, G, C. Adenine, thymine, guanine, cytosine. These are the 4 bases which are forming the skeleton of the DNA. These are the 4 bases forming the skeleton structure of the DNA. We will not go into very details about these things, just know this much, that DNA is composed of 2 purines, 2 pyrimidines, nucleotide bases. These are A, T, G, C, okay, bound together by bonds. So with that, we move on to the next slide. Yes, the discovery of nucleus. One thing we already know, that it was Robert Brown. Robert Hooke, he was different, okay. This is Robert Brown. Robert Hooke had discovered the cell, okay. Robert Brown, this person had described for the first time nucleus in 1831, and he had noticed this nucleus in the epidermis of orchids. In the epidermis of orchids, he had noticed this nucleus. So from where does the word nucleus come? It has come from a Latin term which is meaning seed or kernel. So if we look at the picture over here, just like in an avocado seed, we have the seed is in the fruit, the seed is just in the middle, right? Similarly, in a nucleus cell also, the nucleus lies just in the center, just like the seed of the fruit. So that is why this is an important structure which we will study in details. So who gave the name chromatin? We have read a little about chromatin. I think by now you are understanding the difference between chromosomes and chromatins. The term chromatin was given by a scientist, Fleming, right? It was given by Fleming. From where does the term chromatin come? What do you mean by chroma? Chroma means color. So this chromatin is a structure which takes up or absorbs certain stains or dyes, right? So that is why it has come to be known as chromatin. With that, we move to the next slide. See, what are the features of a nucleus? Just remember, it is the largest organelle which is found in an animal cell. With a size of average 6 micrometers, it is covering 10% of the total cell volume. I repeat, it is covering 10% of the total cell volume. So it is a large organelle. I have already said in my earlier classes, it is referred to as the brain of the cell. And why? Why it is known as the brain of the cell? Because all the functions are coordinated, okay, from the nucleus. All the functions at the cellular level, even at the level of the human body, starts with the nucleus. Okay, that is why it is known as the brain of the cell. And you already know that it is the center which contains all the genetic information in our body. You may ask me a question that how many nucleus is present in a cell? Usually 1 nucleus is present per cell, but there are exceptions. For example, in red blood cells, okay, red blood cells do not have nucleus. Mammalian red blood cells, mammalian RBCs are devoid or lacking nucleus. They don't have a nucleus, right? Then there are some cells which have more than 1 nucleus. For example, the osteoclasts. What are osteoclasts? Osteoclasts are cells which are responsible for resorption of bone. Okay, there are some cells, osteoblasts, which are responsible for bone formation, and there are some cells, osteoclasts, which are responsible for bone resorption. So these are multinucleated cells. Osteoclasts are multinucleated cells. Remember this much. And we have already studied that in case of prokaryotes, there is no true nucleus, right? Because they lack this nuclear envelope, this nuclear membrane is absent. That is why this is known as what? This is known as the nucleoid, meaning like a nucleus but not a nucleus, like a nucleus. Okay. So with that we move on to the next slide. See, here I have just tried to explain that why is it that in some cells we don't have a nucleus. For example, the RBCs. We know that the shape of the RBCs is biconcave. So, because of the absence of the nucleus, the shape can be biconcave. Number one. And number two is because the shape is biconcave, it is able to transport the maximum amount of oxygen because it is able to have the maximum amount of hemoglobin. So basically the presence or absence of nucleus is somewhat dependent on the functions that the individual cells are performing. In case of RBCs, because of their function, there is lack of nucleus. Now let's come to the osteoclasts. In the osteoclasts, as we can see in this picture, there are numerous nuclei within one cell. I think you can see this, right? So why? Why is it so? This is because, I have already said, because of the resorptive activity, okay? Next, if we look at the white blood cells, this is the white blood cell. The white blood cell has multi-lobed nucleus. It has one nucleus, but that nucleus has various lobes, so it is multi-lobed. So these are the various morphological variations which we find in the nucleus of a cell. Next, moving on to the nuclear membrane, also known as the nuclear envelope. It has two coverings, inner, outer. I'll explain with this diagram. See, the outer covering, okay, that is continuous with structures like these. These are the endoplasmic reticulum, specifically the rough endoplasmic reticulum, this one. Okay, and the inner membrane is this one, which is in contact with the nucleoplasm, right? So two membranes, outer and inner. Inner one is in contact with the nucleoplasm, outer one has undulations attached to it, which are known as, we have already read, endoplasmic reticulum. Now, in between the inner and the outer nuclear membrane, we have a space. What is that space? Perinuclear space. This white colored space in between, this is the perinuclear space. So you can ask me, what is the main function of this nuclear membrane? It separates the genetic material within the nucleus from the cytoplasm outside the nucleus. That is the main function of the nuclear membrane. I would like to specify over here that we are seeing some red colored dots in between the continuous membrane, right? For example, we are seeing dots, right? Discontinuations. They are the pores. They are the pores which are present in the membrane. We will go into their functions in detail in the next slide. So moving on to the next slide. What are these nuclear pores? These nuclear pores are basically channels which are present in the nuclear membrane. And what is the main function? The main function is obvious, very obvious. It is movement of molecules from the nucleoplasm into the cytoplasm and from the cytoplasm into the nucleoplasm, right? So what things move? RNAs move, proteins move. So basically these things move in and out from the nucleus into the cytoplasm and from the cytoplasm into the nucleus, right? You already know about the nucleoplasm. It is a semi-fluid matrix, okay, which contains all the chromosomes and it serves as a seat for the various organelles which are present inside the nucleus. That much you know. For example, we have the chromosomes, the chromatins, the nucleolus, etc. That is the nucleoplasm, the matrix. Next, moving on to the next slide. See, I have said that our genetic material is present in the DNA, that is the deoxyribonucleic acid. So that is the chromosomes basically. So here I have elaborated one chromosome. See, in this chromosome, this is one chromosome, right? We have two identical arms. They are the chromatids. Don't confuse. We have studied chromatin. That is the loose form of chromosome, the disorganized form of chromosome. That is the chromatin. And this is the chromatid. That is TID, right? Chromatids are the arms of the chromosome. Okay. And just remember another thing, that if a question is asked that except the nucleus, where else do we find DNA? The answer is mitochondria. Yes, specifically we find it inside the nucleus, but there is one particular organelle where we find DNA that is outside the nucleus. That is the mitochondria. Remember this. Moving on to the next slide, a very important thing that I am trying to explain to you from the beginning of this session, chromatin and chromosomes. So, what is chromatin? Chromatin is basically the unwound DNA. That is the uncoiled form of DNA, right? And what is chromosome? That is the tightly packaged form of DNA. Now a question can arise that when do we find chromatin and where do we find chromosomes? So when a cell is in the resting phase, that is, it is not replicating, okay, it is not undergoing any kind of division. I think you know that a cell undergoes cell division and then the cells multiply to form a multicellular organism. That much is clear, right? So when a cell is not undergoing any kind of division, that is the resting phase of the cell. Now in the resting phase of the cell, usually we will find chromatin. But when a cell is becoming ready to divide, when it is in the actively dividing phase, then we will find the chromosomes. This much is clear? Okay. Now what is their composition? Chromatin is basically made up of DNA, it is made up of histones, it is made up of some non-histonic proteins. DNA, histones, non-histonic proteins, that is chromatin. What is chromosome made of? Chromosome is made up of DNA and protein, right? So, one important part, how many pairs of chromosomes are present in a cell, or how many pairs of chromosomes are present in a nucleus? It is 23 pairs. So 23 pairs means 46. So 46 or 23 pair of chromosomes are present. Remember that much. And another thing we will study in the next slide is that in case of chromatin, there are two varieties of chromatin. Two types of chromatin. One is euchromatin, one is heterochromatin. Two types of chromatin, euchromatin and heterochromatin. What are they? We will discuss in the next slide. So, moving on to the next slide. Yes, just have a look at this picture. What are you seeing? You are seeing some coiled structures and some uncoiled structures. So, these coiled structures, that is the heterochromatin, and these uncoiled structures, they are the euchromatin. So, there is some basic difference in their structure, right? The heterochromatin is a highly densely packaged form of genetic material, whereas the euchromatin is the unwound part, the uncoiled form. But both are varieties of chromatin. They are not chromosomes. Both are varieties of chromatin. I repeat, okay? So there are some dyes which, when we dye a chromatin and watch it under a microscope, we see this heterochromatin part to be darkly stained, whereas this euchromatin is lightly stained. That is because mainly of the packaging. Okay. Next, moving on to the next slide. See, by now you have understood that this X-shaped structure is nothing but a chromosome, right? Okay. Now let me make you understand. See, at the center portion we are seeing a thing which is known as the centromere. We will go into details later. Just know this, that the centromere actually divides a chromosome, okay? Now, see, a chromosome has a central portion which is near the centromere, and a tail portion which is away from it. So depending on the location of the centromere, the whole chromosomes are divided into 4 types. So what are they? When the centromere is just at the center, this first one, it is metacentric, right? When it is slightly away from the center, it is submetacentric, right? When it is near the tail, it is acrocentric. And when it is just at the tail, then it is telocentric. So I think this is quite easy. By the names only you will be able to understand. If it is in the middle, that is M for middle, it is metacentric. So M for M, middle for metacentric, right? And if it is near the tail, that is telocentric. So T for T. So metacentric, submetacentric, acrocentric and telocentric. Now let's move on to the next slide. Yes, the nucleolus. The nucleolus is basically a spherical body which is present inside the nucleus, and it is not a membrane bound organelle. Remember, it is not membrane bound. Okay, so because it is not membrane bound, all the contents of the nucleolus remain scattered within the nucleoplasm. The contents of the nucleolus remain scattered in the nucleoplasm. What is the main function? The main function is active ribosomal RNA synthesis. Main function of nucleolus. What is the main function? The main function is active ribosomal RNA synthesis. Usually how many are present in one nucleus? Usually 1 or 2 are present per nucleus, but maybe more. Okay. So what are the functions of a nucleus? We have studied the parts, we have studied the various functions of the parts. So if we just provide a gist of the overall thing that we have studied, we can understand what the function of a nucleus is. First important thing is it regulates all the hereditary traits in our body. What does that mean in common language? For example, we have features like our parents. Maybe I look like my mother. Maybe I have hair like my mother. So how does that happen? That happens because of this chromosomes, because of the genetic material, because of the DNA. Okay, parts of them are transferred from the parents to the offsprings. Okay, and that is why we look like them. So the most important part is it regulates the hereditary traits. Then it controls protein synthesis, it controls growth, it controls cell division. Protein synthesis, nucleolus. You remember protein synthesis happens in the nucleolus. Then growth. Why? Because it divides, right? Then cell division. Yes, it divides. That is why. Differentiation. Okay. So it has all the hereditary material in the form of DNA strands. You understand this, okay? They are responsible for process of transcription. These processes we are going to study later, because everything together you will not be able to take in. So just take in this much for the time being. Also, it helps in exchange of RNA in between the cell and the nucleus. We have read this because of the presence of the nuclear pores in the nuclear envelope. Yes. So these are the basic functions which are performed by a nucleus, right? Moving on to the last slide. So for the amazing fact for today, just remember that the single human cell has approximately 2-meter long thread of DNA, which is wound together in such a way that it forms the chromosome. 2-meter long, remember this. And for the next class, we will be studying mitochondria. This mitochondria is again a very important cell organelle in the eukaryotic cell. We will be studying that in details too. Till then, take care, stay safe, and if you like this video, please like, share and subscribe, and please comment in the comment section below and let me know how I am taking your classes. Are you able to understand? Is there anything more that I can tell or I can teach? Is there any other way I can communicate with you so that you will be able to better understand how I can improve these things? So please suggest and let me know. Till then, take care. Bye.